Okay, you guys, I'm going over chapter 11. The concept of action potential is the main thing, but in general, we'll be talking about the neurophysiology and the nervous tissues. Let's go over, overall outline and organization. First, this is chapter 11. So what I'd like to do is talk a little bit about the overall organization. How the nervous system organize and work the way that they do. Then the main point of this particular chapter is the process of action potential. Now, in the past, you learned about the neuromuscular junction. We mentioned how the action potential comes in from the nerve and transferred to the muscle. But we didn't really talk about how does it generate, how does it propagate or move within the nerve and causing the neurotransmitters to be released. So this part, the action potential, we will take a look at a few things. One is the type of channel that are involved. In the process of action potential. We will also take a look at movement of the ions. Furthermore, we will take a look at the phases. In particular, we will take a look at resting potential. We will take a look at depolarization. These two, you guys have some familiarity with it because we discuss about depolarization and repolarization in muscles, if you recall. And then finally, we will take a look at hyperpolarization. All right. Along the way, we will also look at a few concepts. Now, one of the concepts of the action potential that we will be discussing is, and it's important in action potential, is why inside of the neuron is negatively charged. Another concept is why action potential is unidirectional. In other words, why is it that it's going in one way and not the opposite way? So those are also the key questions that we wanted to kind of like get the idea and understanding of it. All right. So this is pretty much the outline of what we're going to be discussing right now. Let's go ahead and take a look at the nervous system. The organization is simple. Basically, it's a command center. It's a communication. We already learned how the nerve can communicate with the muscle. So this is no any different. There's always communication. If you're thinking about the five senses, constantly there is a sensory input. In other words, certain stimulation occurring inside and outside of the body, and the body should sense of it, be aware of it. Whether it's the temperature changes, say you have a fever, for example, that would be from the inside of the body. Say, for example, is a hot day today, all right, that would be the outside of the body, but you're sensing the temperature, right? Again, think about all your senses: touch, hearing, vision, taste, smell. All right, so those are all sensory inputs. All right, so in order to sense things, you need to bring in this type of signals into the processing center, which is also known as a integration. In other words, what's happening is, now that you're sensing things, you wanted to get a feel for uh, how do we actually, what does it mean by smell something burning? What does it mean by a red light, for example, or the light is red, okay, or is green while you're driving, all right? So these are interpretation, okay? And the next one is motor output. Motor output, they respond to the stimulation by activation effector organs. And we discussed this already. Effector organs would be like, let's say, a skeletal muscle, all right? You see a bright light, all of a sudden, your pupil is going to contract. Your pupillary muscles are the effector cells, effector organs, I should say, all right? So these are the motor output. Now let's go ahead and take a look at the overall picture again. See if we can make a sense of it. This is just an example. Say, for instance, you're looking at, a glass of water, and your thirst center also activating, your vision and so on is activating via the optic nerve. All that information is going to go to the integration center, which is in this case brain, but brain and the spinal cord is part of the central nervous system. So central nervous system comprised of brain and spinal cord. Okay. So information needs to get there via the sensory input that occurs. So one thing that you need to understand that the sensory input is takes information, right, whether from inside or outside of the body, towards the central nervous system, which is the integration. Most of the time, this is also known as sensory neuron. All right, now what? Well, there are different part of the brain that does different things. You learn, for example, in occipital lobe, that's the visual association cortex. In temporal lobe, you have the hearing sensation. So I'm not going to go over the specific part of the brain because that's what you did in anatomy, I suppose. Um, I wanted to more focus on the neurophysiology stuff. But information sometimes, once it gets into the central nervous system, might need to transfer from one area of the central nervous system to another area of the central nervous system. All right, so for that, you require what's known as an interneuron. The job of interneuron is to transfer information from one part of the central nervous system in another one. So again, think about all the nerves that are inside of the brain, they're communicating with one another, whatever they are, and so on. So these are supposed to be nerves communicating with one another. But information needs to get out. In other words, while you process, okay, the glass is full of water, you're thirsty, you process that, that's your integration system and so on. Now what? Now we need to do something about it. So your effector organs are activating. In this case, your muscles and glands would be the effector. So this is a muscle that would be the effector organ. And the information is getting out from the CNS to the effector happen to be motor output. Motor output always take information from the CNS towards the effector organ. A lot of times these are done by the motor neuron. Motor neurons do that, and you're already familiar with it. When we discussed the neuromuscular junction, that was right here. That was association of the motor neuron with the effector organs. So this is overall ideas. Again, it doesn't have to be only for vision. You could think about another and so on. So make sure that you understand the concept. For example, you know, you're driving your vehicle, all of a sudden you're hearing a siren from, you know, paramedics, for example. And again, same ideas. Now your hearing sensation is the sensory input. This information is going to go to the brain. Your brain is going to process, oh, this siren means emergency. I got to pull over to the side. And again, via the motor neuron, you will probably stimulate the effector organs of your calf muscles or leg muscles, and you press down on a brake. All right. So again, in this case, I was not a vision, it was hearing. So again, think about your other senses as well. And these are all part of the overall picture. All right. So moving right along, if we could go ahead and organize a little bit further and understand what's going on, is that there are two systems. One is well, actually there's more than that, but central nervous system, which is the brain and the spinal cord. That's a command center. That's the integration where interpret things, making sense of things, like we make a sense of red light being making or hearing the siren means to stop and so on. But then there is also a peripheral nervous system. Peripheral nervous system basically is all the nerves outside of the central nervous system. So if you exclude brain and the spinal cord, which was part of the central nervous system, we mentioned that was a brain and a spinal cord, everything else is a peripheral nervous system. This includes cranial nerves, this includes the spinal nerves, messages comes in towards the CNS and getting out of the CNS, all of that is peripheral nervous system. So if we go back and take a look at, you know, if we take away the interneuron right over here, okay, where the integration, everything else is peripheral nervous system. So this sensory neuron is part of the peripheral nervous system, and over here is a peripheral nervous system. Information going, information coming out. All right. So that's the idea about the organization, really. But there is another system that later on I will discuss, and is mostly involved in autonomic nervous system. So if we go ahead and take a look at autonomic nervous system, we might be able to just touch upon it. And then this is the topic of chapter 14 we're going to look at later on. But two systems of sympathetic and parasympathetic are important. And the effector organs are kind of like different. Instead of a skeletal muscle being a effector organ, which was the case in the sensory nervous system or somato nervous system, SNS. In this case, being what's being regulated or effector organs would be smooth muscles, cardiac muscles, and glands. Again, autonomic means automatic. So these are kind of like we don't have much of a regulation that is under our voluntary or conscious control. In other words, if you exercise, your heart rate automatically is going to go up, right? You won't be able to stop that when you're exercising, right? So when you're resting, the heart rate is going to go down. So as a lot of the smooth muscles that are involved in our digestion. When you're digesting something, you cannot, like, tell your smooth muscle to stop from digesting or processing food, and don't take the food from the stomach to the small intestine. It's just going to happen. The smooth muscles are going to contract. As you know, this is known as peristalsis. And again, what happens is the food gets processed that way. All right, so just a quick note, going back to the earlier our discussion about the peripheral nervous system and so on. So we discuss about the afferent division or sensory division. These are sensory neuron. So there is a afferent, and then there is a motor or efferent division. So motor neuron that we discussed coming from the CNS to the effector organs, as is explained right over here, right? But efferent is another classification, or just saying that information that gets out. So sometimes instead of sensory division, we call it afferent division. So afferent has an A. So you could think of information or signal arriving where they arriving to the central nervous system. And efferent division, E, you could think of that, impulses exiting, exit, all right, from the CNS. Now, sometimes this signal could be coming in from our organ. And if it is our internal organ, usually we call it visceral afferent fibers, as opposed to sensory afferent fibers. Sensory afferent fibers, the impulses can be coming from the skin, from joints, from ligaments, from the skeletal muscle, things in that nature. Visceral, they'll kind of like, think about your deep organ. Say, for example, you have a tummy ache. You still know you have a tummy ache because the information went from your, nerves surrounding your stomach within the body. So that would be visceral, or also known as kind of like a deep organ, sort of to speak. So I just wanted to mention that. But regardless, let's go ahead and take a look at really quickly a little bit of anatomy of the neurons. I don't want it to kind of like go over too much detail because you're supposed to have that already, but it won't hurt to refer to the different parts because we're going to be talking about the nerve. So the nerve, usually known as a neuron, all right? Neurons means nerve cells. And let's go ahead and take a look at the parts. So this part is called dendrites. There's a lot of processes and so on. Now, what you need to know is that these dendrites are the site for the receiving the signal. So this could be another nerve. I'm going to draw it as small, but it could be another nerve right over here, and the signal that they're sending can be received from here, all right? Or there could be another nerve right over here, and again, the signal can be received from here. So all of these are different signals that they can get received and so on. These are collectively known as dendrites, all right? Receptive region. All right, next up, we got cell body. If you look at it from a distance away, majority of the cell is this part right here, which is in yellow area. So basically, this part is also known as soma. Soma means cell body, okay? So what's happening in the soma? This is the topic of chapter 13. Actually, I think it's chapter 11, part B, part 2. But we'll discuss that a little bit later on. But information kind of get processed in here, all right? And it might further send its signal to the next region, which is the axon hillock. All right, axon hillock is a funnel-shaped structure and is actually important. But before we go on to the important part of the axon hillock, there are a lot of organelles, endoplasmic reticulums and so on. Sometimes this is referred to as nasal body. There's nucleus, we already know, that contains the genetic material and all that stuff. But let's go back to axon hillock. Axon hillock is this funnel-shaped area right here. Right? You can see it resembles a funnel right here. Okay? But right on these axon hillock, there are numerous voltage gated channels. We'll talk about different type of channels, but I think you already know what is voltage sensitive gated channel is. So I'm just going to refer that in here and put in sodium voltage sensitive. Sodium voltage sensitive channels are numerous right here on the axon hillock. Then you have a long axon. So most of the axons have been covered, but if you look at between the Schwann cells, you might be able to see the axon here, also axon here, also axon here. Sometimes if it's between the myelinated sheets known as Schwann cells, or sheets of Schwann's, it's called the nodes of Ranvier. But again, impulses will go towards this direction. This impulse direction is unidirectional. In other words, once the signal receives, then again, it's a certain process occurs in the soma, and if the signal needs to continue going, which is action potential in this case, is going to fire, and we'll discuss that, and this is the direction in which it's going. The rest of the story, you probably know more, or you can recall it. This part right over here is called axonal terminal. The axonal terminal happen to be where the neurotransmitters are being released. All right, so you have some ideas about this already. Okay, so this is just a general anatomy of it. What we would like to do now is take a look at the type of channels in different areas. I think that might be important to know. So in other words, if we go ahead and take a look at our organization, we're pretty much done. We discussed CNS, we discussed PNS, we discussed autonomic nervous system, we discussed how sensory integration and the effector are coming to a place, and motor neuron, I forgot about that, and then effector. And then we discussed a little bit about the anatomy of the neuron. So maybe I should have included, neuron parts or anatomy review. And now we are going to go over the type of channels that are involved in action potential. So one of those channels I already mentioned, that would be the sodium voltage sensitive channel. So again, this happened to be on the axon hillock, but what other channels can we observe and where they are located? Well, I think you can recall that on the axonal terminal, all right, you can pause the video and see if you can recall what type of voltage sensitive channel we had in there. We discussed it in neuromuscular junction. In other words, action potential comes in and then it does something, all right? Well, opens up certain channel, all right? Do you remember what it was? Okay, congratulations if you got this. That's calcium voltage gated. So we can have calcium voltage gated channels right on the axon terminal, all right? But what about towards the axon right here, for example? What kind of a channels can we see along the axon? Pretend you didn't have these myelinated sheets. We'll discuss the role of it maybe towards the end, all right? But if you didn't have it, what other channels would you see on the axon, all right? So on the regular axon, say for example, right over here where the axon hillock is no longer there, axon hillock, you can only see sodium voltage sensitive. But on the regular axon, maybe I should use a different color. On axon, we will have sodium and also potassium voltage gated channel. When I say voltage gated channel, that means they're sensitive to voltage changes. All right. So can we see these voltage gated channels on anywhere else? And no, we cannot see it in soma. We cannot see it on the dendrites. These are the where we can find it. All right. Now, let's go ahead and draw a neuron. So here I have a neuron with a long axon. So right now I'm going to go over here, okay, and show you basically what is the sodium voltage gated channel looks like. These are sodium voltage gated channel, folks. All right, pretend this is axon hillock. All right, so I'm just showing you that there are numerous, okay? You could put in more of these around here, okay? So there are literally more of them around. I just don't want to put them. But now we are going towards the axon. So this part right over here is the part of the axon, no longer axon hillock, like the funnel shape that we discussed. So what do we see in here? Well, we see more sodium voltage gated channel. And also we'll see some potassium voltage gated channel. Pretend these are type of protein, all right? So all along the axon, we can see these potassium voltage gated channel as well, okay? So there you go. And this continues. All along the axon, you have these sodium and potassium voltage gated channels. All right, and all of them are closed, right? These are sodium voltage gated or sensitive channel, right? The one with red, and axon hillock, only you have that. And again, I would draw a lot more to kind of like illustrate that there are so many of them and they're all close. All right, so then you have potassium voltage gated channel right here. All right, so those are the main channels, obviously. And then again, on the axon terminal, which I cannot show it to you. Well, actually, I might be able to show it to you, and that would be right over here. We mentioned that you have the calcium voltage gated channel. All right, we're going to come back to this. All right, but what other channel can we see, and so on? Well, all along the way, and it doesn't matter where, like for example, if this is being the dendrites or on the soma, I'm running out of room, and I would like to maybe draw this separately so you could see it a little bit better. If we go ahead and take a look at the neuron, All right. These are the dendrites. These are the soma. This is the axon hillock. This is the, axon. All along the way, there are sodium-potassium pump. All right. So we just take a look at any of these, they're all sodium potassium pump. So if I just like look at this part right over here and take a look at this part right here, that would be the sodium potassium pump. And this would be inside of the cell. This would be outside of the cell. Again, we're just looking at here. This is in, this is out. So you're looking at pretty much this section right over here, for instance. All right, I just enlarge it. And if you recall, sodium potassium pumps, how many sodium out and how many potassium in? This was the case in muscles or anywhere else. So think about that. If you were able to figure it out, great job. 3 sodium out and 2 potassium in. And if you recall that this pump is an active transport, so it needs the ATP from the mitochondria. Mitochondria generates the ATP. ATP can break down into adenosine diphosphate. The phosphate bonds are having a high energy. So when you break it, you release the energy, and this energy is enough to pump in 3 sodium out for 2 potassium that comes in. All right. So that's the job of the sodium potassium pump. And guess what? We have it all over the nerve, on the dendrites, on the soma, on the axon hillock, on the axon, on the synaptic terminal or axon terminal. So sodium potassium pumps everywhere. They're non-stop working. Always think about like you are in a boat and there's like a leakage somewhere and the water keeps coming in and you got to get the bucket and keep pumping or taking the water out and pump it out continuously. You do not stop. If you do, your boat will drown. Okay, will go towards the bottom of the ocean, for example. You will sink. All right. So that's really the idea of that. Okay. Now, so that's another channel that was important. Now, next channels we refer it to as leak channel. Leak channel are actually very much they're passive channel, so there's no energy required. Therefore, they're considered to be substances moving from high concentration area towards the low concentration area, so it's favorable. But there are various leak channel on our neuron as well. When we go ahead and take a look at the neuron, all right, and take a look at this part, for example, of the neuron, okay, so we're just kind of like showing a little bit bigger, and this would be inside of the nerve, this would be outside of the nerve, and we have leak channels. So the leak channels that we do have are for sodium. So notice that I'm going to leave this channel open, pretend this line was not here. All right, I could erase it a little bit later on, but these are the leak channels. So the leak channels happen to be here, you're looking at from the top view. All right, so you'll see the opening there too, that is open right here. This opening is the same thing as this opening. All right, so there are various leak channels. All right, this is called sodium leak channel. So again, if you recall, we discussed facilitative diffusion. Now, because of the sodium potassium pump earlier we discussed that the sodium potassium pump can pump 3 sodium out and bring in 2 potassium. And because this is driven by the ATP and is not favorable, it takes energy. So if these pumps are constantly working and you pumping 3 sodium out and bringing 2 potassium in, and everywhere these pumps are located, 3 sodium out, 2 potassium in, well, what does it do? Well, what does it do is that outside you're going to have a lot of sodium. That's what's going to happen. So higher sodium concentration happen to be on the outside. So pretend these dots are sodium channel. I'm sorry, sodium ion, cation. All right. So you'll see a lot of sodium on the outside. All right. Furthermore, because of the sodium potassium pump, is working, then inside you will have higher amount of potassium. So this will give me a higher concentration of potassium inside of the cell and lower concentration of potassium outside of the cell. All right, so far, this is due to the sodium potassium pump. But if you notice, I'm putting some potassium outside as well, and I'm putting some sodium inside as well. This is due to the potassium and sodium leak channel. So this is potassium leak channel. So what it does, once the potassium, it goes inside, what happened is some of these potassium, they could just leak back out. All right. And same thing with the sodium. Now, before we put sodium, there are actually more potassium leak channels. So right now I'm putting less. So it would be a good idea to put in more of these leak channels. All right. So leak channels, they're always open. So I should put in more of these leak channels. Right. They're always open so potassium can leave. Right. Pretend that they are open and the potassium can leave. So compared to the number of the sodium, you actually have more potassium channel open. And so more potassium can leave, all right, through this, the non-voltage gated, they're always open, leak channel. Facilitative diffusion was the process that we discussed. So sodium is keep leaking out, all right. Now, I like it a little bit better because as you can see, I have 1, 2, 3, 4, 5, 6, 7, 8 channel under this drawing and so on. Now, as far as the sodium goes, the sodium will go the other way because again, you're going from high concentration to the low concentration. Inside was a low concentration, all right? Why? Because 3 sodium was pumped out. So now sodium can leak in too, all right? But not a whole lot because the numbers are limited, all right? So less sodium is going in. The point is this leak channel is always open, all right? And the cell can make lots of different type of protein. In this case, there are transport protein. Again, we discussed how you could use the nucleus, all right, and make the DNA inside, make the RNA inside of the nucleus. That would be the transcription. Using the translation, you can make the protein. So these are type of a protein. We discussed these are membrane protein. So again, a lot of things that we discussed earlier on, it's coming alive right now. Okay, so so far we looked at voltage gated channel, we looked at sodium potassium pump, And we also looked at the leak channel, all right? So that's what we covered so far. There are more other type of channels as well. And let me just briefly talk a little bit about that, and then we'll come back to what is happening here, all right? Another type of channels that you might be able to see a little bit later on happen to be chemical gated channel, which again, you're familiar with chemical gated channel, all right? A chemical gated channel happen to be on the dendrites and on the soma, cell body. So if you have a particular nerve, all right, and the dendrites and all that is the long extension, the receptive parts, and the soma, and everything. So we go ahead and take a look and enlarge this part, all right? You see something like this, for example, all right? And you could have chemical gated channels in there. Chemical gated channels are closed. Or if we go ahead and take a look at this part, for example, same thing. There could be chemical gated channels in here as well. So chemical gated channels, they're normally closed. All right. They will be open when there are particular neurotransmitters attaching to them. Then they can get open, for example. All right. So on this case, I'm showing this channel being open because of the neurotransmitter. Some of these neurotransmitters you're familiar with was acetylcholine, but we looked at this type of channel on the muscle itself. Well, guess what? They could have it on the nerve as well. But you're not going to have it on the axon or axon hillock or axon terminal. You only will see it on the soma and the dendrites. That's another type of channel. All right. Furthermore, if we go ahead and take a look at other type of channels, so I'm just going to maybe erase this part. And there are certain channels that can open and close a little bit differently due to internal activation of the neurons, and we'll discuss those a little bit later on, okay? But those are the main parts. I think it can get you going. Let's come back to this part right here. Now, we still haven't gone to the process of action potential yet. These are the type of channels that are involved in the process of action potential. So that's why I went out and talked about it, okay? If we go ahead and take a look at the nerve itself, and you put your voltage meter in there, all right? And you place it anywhere. You could put it in the axon, you could put it in the axon terminal, whatever. It would read about -70 millivolts. -70 millivolts. This is our neuron. Again, if you work with the other neurons or you're looking at different kinds of neuron and so on, it's around there, but it may be, -65, -73. It really depends on the type of neuron and which organism we're looking at. But for the majority, our neuron is -70 inside. So if you put in the voltage meter in there, it will show you the voltage, all right? Kind of like your battery. If you put your voltage meter in there, it will give you the voltage to see if your battery is dead or not. Surprisingly, it's giving us a -70 millivolts. This is called a resting potential. Resting potential is at negative 70 millivolts. That means there's nothing really firing. There's no signal is coming in or being processed or anything like that. And this is called a resting potential. Resting means like they're not firing. So why is it inside that is negatively charged? All right. So I'm going to go over three particular reasons why inside is negative. In other words, we want to know why is inside is negatively charged. Why should it be negative? Why not positive? All right. So three reasons for why inside is negative 70 millivolts. Number one, there are protein molecule inside of the nerve. Proteins, as you know, they're large. They cannot pass the membrane. So if we go ahead and draw these proteins as such, they cannot pass and get out of the membrane. These proteins are negatively charged, all right? So proteins inside of the neurons are negatively charged, all right? So again, chain of amino acids, some of the amino acids, they have negative charge on them, so therefore the protein is going to be negatively charged. Number two happen to be there are more potassium leak channel. So earlier we mentioned, I don't know, there. So remember, the blue ones were for potassium leak channel. And again, when we draw it, we draw more of them compared to the red one, all right? So it looks like we got 5 red channel in this case. 1, 2, 3, 4, 5, 6, 7, 8 of them. This is not really a correct proportion. Just know that there are more potassium leak channels. So if there is more potassium leak channel, rather than just me memorizing them, what does it do? I mean, more potassium is going to be leaving. That's what is going to happen, because outside you have lower amount of potassium. So more potassium is going to be leaving. Potassium is positive ion. When more positive is leaving the cell, what's going to happen inside? Inside is going to be less positive. Or in our case, more negative is the same thing. Positive ion is leaving. All right. So if it's leaving, then inside of the neuron is going to have, less charges. In this case, negative 70 millivolts. Number three. Third reason why inside of the neuron is negative happen to be sodium-potassium pump. We have these pumps all over. So all of these three, why inside of the neuron is negative. All right. So sodium-potassium pumps 3 sodium out as opposed to 2 potassium in. So again, it pumping more positive ion out. So it makes sense for the nerve inside to be negative more and outside be positive. And they are, this positive and negative, they are separated by the membrane. Same thing in here, you are positive. What does this remind you of? One side is negative, the other side is positive. Battery. Battery have one side is positive, the other side is negative. What is inside your battery? Potential energy. How can you use a battery? Well, again, when these charges are floating, right? And basically the charges changes, and that is energy, is a stored energy in the battery that allows you to turn on your flashlight or use your phone or whatever the case might be. So same thing in here. This is a stored energy, which later we will find out how this ions, they could go move in and out, which the charge is going to be different now, and that causing the flow of the current and the voltage to keep changing back and forth along the axon, which is known as, you guessed it, action potential. All right. So those are the three reasons why insight is negative. We don't want to just memorize things and not knowing why. We wanted to kind of like take a look to see what is happening, and basically we just establish that as well. All right. Now, let's go ahead and take a look at one more thing. Okay, so now that we watched the video clip, and there are many other clips on the YouTube, you guys can look at different ones if you like. But right now, we learned why inside of the nerve is negatively charged. But let's do something else. Let's say that here is your neuron, and you put on the electrode in here now, all right? And let's say you're zapping this neuron with certain voltage, and obviously you could, put it at a certain voltage, for instance, in here, and this happened to be, plugged in. So you're getting the electricity and you're zapping it with certain voltage. You're putting the electrode in there and you're zapping this. Okay, so if we zap this and set a voltage at particular volts, and by the way, we can still measure, we could put a voltage meter in here. Right now, if I don't zap this nerve with any particular volts, it's going to read -70 millivolts. We already mentioned that, all right? But what if I zap it with, let's say, +5 millivolts? Then what will happen? So this voltage meter is going to change probably to, can you guess? +5, -70. So now I'm going to be having a reading of negative, if I started 70 minus 5, that would be -65. All right, -70 plus 5, that will give me -65 millivolts. All right, so but pretty soon, and again, if I have a voltage meter in here, all right, this one still reads -70 millivolts. So what just happened is that the voltage change only locally, only around here. As the distance pass away, or you're going more distant away from that, the voltage doesn't reach here, all right? In this case, it's still the same. So this is called graded potential. Graded potential decays with distance. In other words, further down the road, it won't affect it. Now, why wouldn't it affect it? Because it's not firing, it's not keep changing and going to that areas and so on. Keep in mind, very soon, this -65 is going to read -70 again. Why? Because you only zapped it one time, and sodium-potassium pump will restore the balances. Again, it can pump up more positive ion out as opposed to potassium coming in. So again, it restores it back to -70. Now, if I zap this at +10, similar thing is going to happen. In other words, in here is now is going to read -60, and again, that's not going to change either. All right. What if I keep trying and zapping it with +15 millivolts? Now something else is going to happen. All right. So in other words, this -70 all of a sudden is going to change to around +30 or +35. All right, so all of a sudden this one is going to change from -70 to around +30, 35. Okay, which is a little bit different now. All right, this is +35. So this is very interesting because when you're looking at it, all right, this +30 or +35 is now, let's put 30 to 35 millivolts is changing, right? And very soon, you'll see that my second voltage meter down in the axon also reads +35, all right? So there might be a delay a little bit because the signal needs to reach in here and then you read it. But very soon, again, this would change. But we're not going to go ahead and take a look at the change. When the inside was negative and all of a sudden becomes positive, this is called depolarization. So this is a classic example of depolarization. We will discuss what happens during this depolarization, but action potential apparently fired in this case, and they are going along with the axon, and it reaches my second voltage meter as well in this case. So if I have a graph and I wanted to demonstrate this in a graph, I could just draw a graph of what is happening. X-axis would be time. This most of the time is milliseconds. and y-axis would be membrane potential. We started membrane potential as a -70. We said this would be the resting potential. All right, so this is resting potential. At right over here, let me show you with a different color. All right over here, we zap. This was our stimulus. In particular, on this case, we zap it with +15, which again, if you zap this with +15 millivolts in our case, then what happened is it might take a little bit later, all right, and it reaches the -55, because +15, -70, that gives me -55. Now, -55 considered to be threshold. This is the threshold. This is the minimum amount of voltage necessary to get the sodium voltage gated channel open. All right. So if you recall earlier, I mentioned to you that there are a lot of sodium voltage sensitive channel on the axon hillock right over here. So when you apply enough voltage, in this case, raising it to the level, the threshold, these are sensitive to, so there's going to start opening. I might have gave you this example in the muscle, I'm not sure, but pretend that there is an automatic door that opens, all right? As you put your foot on the platform, on the automatic door, it opens up because it senses your weight. But there is a minimum amount of weight that can be sensed for the door to open. All right, so again, the door is opening. It's kind of like this sodium voltage gated channel. All right, voltage. And the amount of weight that you put in on the platform, in our case, is kind of like a voltage. All right, when there is enough voltage in there, is going to get this channel open. And if these channels are open, remember, you had higher amount of sodium outside, so they're going to rush in. All this sodium is they're gonna rush in, as earlier we discussed. All right. Earlier I told you there is a higher amount of sodium. Now they're not going through the leak channel now, so we are not there. Okay, we are going at the here. These are the voltage gated channel. All right. So when there is enough voltage, all of these channels are opening. So again, pretend that this part right over here are opening, there are opening, and higher amount of sodium that were outside, they are rushing in. So these channels are opening. This one should be here. Now it's opening. All right. So, and this one is supposed to be open too. Anyways, so this causing inside to be depolarized, because all of a sudden the sodium is coming inside. Now let's analyze why sodium is coming inside to begin with. Well, obviously earlier we mentioned that outside you have higher voltage, positive ion, and inside, due to the three reasons that we discussed, were negatively charged in here. So obviously positive and negative attract one another. When these channels are open and you have higher amount of sodium outside, one reason why sodium are rushing inside is because you have lower sodium inside. Second reason why sodium rushing inside is due to this positive and negativity. Positive and negative attract one another. So this is called electronegativity chemical reasons. Electrochemical, chemical because of there's different chemical, higher sodium outside as opposed to lower amount of sodium inside. And the electro because of the electrical charges are different. So this is called electrochemical gradient, all right? So that's why the sodium is rushing inside. So no longer in here is going to be negative 70 millivolts because now all of these channels suddenly are opening on the axon hillock and the sodium is rushing inside like crazy, all right? So imagine everybody's waiting outside to go to the concert. All of a sudden the concert door opens and say, okay, you guys can come in. Everybody's rushing inside to go to their favorite concert to see and so on, all right? So it's kind of like that. Anyways, no longer this side is going to be negative 70. All of a sudden, due to rushing of the sodium, which is the cation, is positively charged inside, now you're going to have positive 35. At one point in time, these channels are all opening in the axon hillock. We are on the axon hillock right here, all right? When you're reaching negative 55. Negative 55 is the threshold, just like the automatic door that we discussed opens, all right? It opens and then they can rush in, all right? So there's none of these doors that are halfway open. Once it reaches that, they're all open. So again, automatic door are kind of like that. If enough weight, put it on the platform, the door is going to open. You know, it's not like halfway is going to open. If there's less weight or there's more weight, they're going to open fully. No, they're going to open regardless. So that's the idea behind this, okay? So let's go back and find where we were at earlier, right here. So the stimulus came in, that was positive 15 millivolts. Now sodium is rushing inside. So for that, I'm going to show you on the threshold, sodium channels are open. And as a result of that, sodium is rushing inside. So I'm just going to put sodium influx. Influx means coming inside or rush inside. All right. So sodium enters. And this is called depolarization. Where are we at this point in time? At this point in time, we're around 30 to 35. So let's just put 30, I think, to be consistent with your book. All right, so around 30 to 35. Remember this membrane potential, it's in millivolts. All right, so if you want to put in the unit. Now, eventually what's going to happen is when there is enough sodium rushed in, then they don't want to go back in anymore. All right, so where were we? All right, so at one point in time, enough sodium went in. So no longer you're going to have negative 70 anymore. Remember, now we are positive. So we are positive in here, all right? And the amount of sodium outside is no longer high on this side. We don't care about this side yet because we are not there yet. Okay, so sodium is coming inside, all right? No longer you're going to have higher amount of sodium outside and lower amount of sodium inside. So they're kind of like reaching equilibrium, right? And due to the fact that a lot of positive ions rush in and they got attracted to the negative, now outside along this area is going to be negative and inside is going to be positive. So again, the charges are different now, okay? So no longer sodium wants to come in, and these gates are temporarily, they're gonna close down, okay? I'll show you the two gates that are there shortly, but right now, just know that no more sodium is coming in because they are closing, they are closing, they are closing. So at one specific time, you know, when enough sodium went in, then they don't want it to go in anymore, and the permeability for sodium to come in is going to be reduced. That means less sodium is going to come in as you're reaching the positive 30, all right? However, this is enough voltage to do two things. Nearby, these are also voltage gated channels as well. And nearby their voltage gated channel as well. So remember now inside we have positive 35. That's what our measurement of, or positive 30, let's say, right? But what do we need in here to get this one open? We only need negative 55, which is, we have positive 30. Now with distance, this might die off, but they're not gonna die off that much. I mean, negative 55, that's enough to get them open. So again, now we got nearby channel opening, and again sodium from this side is going to rush in, and again causing the positive, same reason, electrochemical gradient, all right, inside to be positive. And now again this positivity is going to open up a potassium channel now. So potassium channel is going to be open, so let's open it, all right, and as a result of that, potassium is going to exit now. Why is the potassium is going to exit? Similar reason. One, you have higher concentration of potassium inside as opposed to the outside. Two, you had positive, or because we say it was depolarized, so you have positive in here, and outside we established that was negative in here. So potassium wants to go to the area that is negative and also has a lower amount. So again, same reason, electrochemical gradient. So potassium is leaving at this point in time. All right, potassium is leaving at this point in time. All right, now, so let's illustrate this part on our graphical presentation that we had earlier. At this point in time, potassium is leaving. This is called a repolarization. Repolarization is when the potassium leaves. Sometimes this is known as efflux, exit, versus influx. So potassium leaves, all right, and up to where? Up to the negative 55, because when the potassium is positive ion, when they're leaving, guess what? I mean, they're going back towards the threshold. In other words, it's going back towards their negativity or the resting potential, all right? So potassium keeps on leaving, all right? However, something interesting about the potassium and just the nature of it is that these channels, they're slow in closing. Think about some of the elevators. They're open, but when you go to the elevator, it takes forever to close, all right? Some elevators are not like that, all right? You press the button and you just wait, and it's closing slowly and so on. So it's kind of like that. Therefore, more potassium have chance to leave. When there is more potassium is leaving because the door is still open, all right, or closing it slowly, what is going to happen is going to cause hyperpolarization. Hyperpolarization when you are below the resting potential. So I'm just going to use a darker color for here, all right? So this happened to be called, all right, hyperpolarization. So let me just put it here. Hyperpolarization when the potential, membrane potential is more negative now. More negative than what? Than the resting potential. Due to more potassium is leaving. But interestingly enough, you'll notice that eventually what's going to happen is you, after the hyperpolarization, you're reaching the resting membrane potential. All right, so this is now resting membrane potential. All right. So how did we achieve that? I mean, there were more potassium leaving. Okay. Again, in order to understand this part, we need to go back maybe and take a look at the channels right over here. So again, the reason why from the hyperpolarization, when the potassium is leaving and there's flowing closing, so hyperpolarization achieved. Why is it going back to the resting potential? Because remember, there were a lot of sodium potassium pumps in here. So again, they do the pumping, 3 sodium out, and they bring in potassium in. So these pumps are extremely important. And earlier I told you, you can find it anywhere. You can find it in the axon and the axon hillock, on the soma, on the dendrites, etc. Basically, what they're doing is they rebalancing everything. Remember, sodium went in, so no longer you have high sodium. So this is wrong now. This sodium is not going to be higher in here as they were going inside. And in here is not going to be lower in here because they already moved inside. So now you have to restore that. And in order to restore, again, sodium is going to be pumped out, 3 sodium out and 2 potassium in. So similarly, the potassium that goes in, it will restore the balance that here extra potassium went out, and the sodium that was pumping out is going to restore the sodium that came in. So when these pumps are constantly working, it restores the balance for you. We also have the leak channels going on at the same time. Leak channels always open, and they are going against their concentration gradient from high to low. So therefore, all of this is at work, and sodium potassium pump is extremely important to rebalance, restore everything so that the signal can go again and the stimulus can trigger the action potential to move right along. All right, so everything repeats. In other words, this positive ion that we achieve, the depolarization, not only is going to open up the potassium channel, but the nearby is going to open up another sodium channel. So again, on this end, sodium is going to move in, depolarization is going to happen in here, and this depolarization is going to open up a potassium channel. So now potassium is leaving, so therefore voltage keep changing. All right, positive, negative, negative, positive, positive, negative, and so on. So this is the voltage changes. This is action potential. This is the current that is passing through. All right, so this is what we detect on our voltage meter. All right, so this is also known as propagation. Propagation means the action potential is moving along the axon, all right? It goes all the way to the end. Over here, it goes all the way to the end, which again, having the voltage changing and going towards the end, finally, they're gonna open up the calcium voltage gated channel, calcium moving in, and then again, releases the neurotransmitter, which is a review for you. So interestingly enough, that's what happens during this episode. So recap of what we discussed. We looked at resting potential, how it's achieved, depolarization, how it's achieved, which type of channels are involved, especially the axon hillock, and then the repolarization, which is potassium is leaving, all right? And again, you're going back to towards the resting membrane potential. And why is it that we get the hyperpolarization? More potassium is leaving. And then again, restoring everything back to the resting potential. So now you could trigger another action potential. Remember, the key is the threshold. Action potential either fires or it doesn't. It's kind of like shooting a gun. All right, if you put your fingers on the trigger point, all right, if you put enough force, the gun is gonna shoot. All right, so there is no halfway in between. It's either it's gonna shoot or it won't. If you put less force, the gun is not gonna fire. All right, if you put in more force, it's gonna fire the same way as the minimum amount of force. So it's kind of like that. The threshold, once it reaches, it opens. All right. So action potential is either going to fire or it won't. Okay. Now you're saying to yourself, why is it that I should care? Okay, fire or it won't. Remember what we opened up the lecture with. We are in here. In order for the sensory neuron to reach the central nervous system, so you could sense things, so you could interpret things, so you could integrate things, and so on, action potential needs to trigger. And electrical impulses, they need to reach in here. Same thing, electrical impulses, they need to reach in here. And throughout the motor neuron, they need to release the neurotransmitter, in this case, acetylcholine. And then again, action potential needs to go to the muscles so they could contract. Everything is dependent on one another. So that's why we wanted to go over the action potential. That's why you wanted to find out that the electrical impulses, they're going from one area of the nerve to another area. Now, we are not quite here yet. This is where the neurons communicate with the signals, with the chemical signal, neurotransmitters. Same thing in here. Same thing in here. In here, you already had it. That was the neuromuscular junction. All right. So we're going to take a look at the second part a little bit later on. All right. About how this communication taking place. So action potential needs to fire. Okay. So we can send the signal to a proper destination. In this case, from sensory to the integration center, and from integration center to the motor neuron, and from the motor neuron to the effector organ. Another thing is you want this information to reach a particular destination in a unidirectional way. What that means not only they need to fire and continue, but also they need to go this way. You can't have action potential halfway, it changes its course and come back. All right. So this is called unidirectional. That means only one direction that you have. It's kind of like a one-way street. All right. You cannot go the opposite. All the incoming traffic is coming in from one way. Obviously, you should know the reason why. I mean, you wanted to send this information to here, nowhere else. All right. So that's why you don't want the information to go back, because then you won't sense anything. All right. If the information goes back, you won't be able to see, you won't be able to hear, you won't be able to taste, you won't be able to touch, you won't be able to detect the temperature, you won't be able to do much. So that doesn't make sense. All right. So interestingly enough, we will discuss why is the action potential is unidirectional. All right. So that's coming up shortly. remember that also, sodium potassium pump restoring the balances as well. And that is something that this video did not include it. Also, I did not talk about the two type of uh, gates. Now, the red one representing the sodium voltage gated channel, all right? But if we go ahead and take a look at earlier, you could see there are actually two gates going on in here. So let me see if I can go back to the video and see, all right? So over here, these are called inactivation gates. Up here is activation gate. So in other words, when both of these are open, then the sodium can come in. But you could kind of like plug it, and inactivation gate, when it's closing that, temporarily is being inactivated. That means now is not working anymore. Even if you apply voltage, it's not going to work anymore, okay, until it resets. But the point is right now, and this is referred to the relative, absolute refractory period and the relative refractory period, which comes up shortly, but basically, they have two gates going on, okay? So that's the idea behind that part, and I will explore that part a little bit later. But let's go back to the video to find out where we were. So again, the sodium potassium pump plays a role as well to balance this, so as the leak channel, and that's where we are. So let's take a look at what happens in the summary. Schwann cell. This is Schwann cell, okay? And they wrap around. So they offer insulation and also the faster transmission. Saltatory conduction is the term. And what it does, it enables the electrical impulses to go from one node to another. That's why it goes faster. They don't have to open up all those channels. In other words, it jumps. It can go faster. Okay, so the video talked about the refractory period. There's two kinds of refractory period. Absolute refractory period is basically prevents the neuron from generating an action potential. During this event, if you recall, we mentioned that the inactivation gate, which was this part right over here, right? This was inside, this was outside of the nerve. Inactivation gate closing, all right? So at this point in time, no more sodium they can get in, all right? But temporarily, this part is now inactivated as the name of it implies, because this is called inactivation gate right here, all right? So they are being inactivated. If you could picture that, for example, some of the elevators again like that, when you enter the elevator and you notice like you press the wrong button, you press like, you know, second floor, but you wanted to go to basement, all right? And you keep pressing the basement, but it's already done, all right? It's inactivated now. You have to wait, and then, you know, you could go to the next floor. It's kind of like that. So it prevents the neuron from generating action potential. Why you want to do that? Because you want to complete the action potential. Remember, the signal needs to reach its destination. You don't want any disruption in that, okay? So, and also it ensures that each signal is separate from one another. The one signal is coming in, is fired, now the second signal could come in. And again, it helps for propagation of the neuron to go one way. So if we have a nerve, for example, right here, okay, when you have a lot of these sodium channels that are voltage sensitive, if this one is inactivated, for example, and this one is inactivated, now pretend that sodium is going in here, so in here is a positive. We mentioned that it's going to open up this one because voltage is changing, but you don't want this to open this channel, because if so, then now the action potential is going to go opposite way. So that's why you wanted to temporarily inactivate it, so you could achieve the unidirectional way. So that's the main ideas. When the inactivation gate is closing right here, temporarily they are being inactivated, as the name of it implies. Is it going to last forever? No. Very soon they're going to reset again, all right? So again, this part, activation gate right here, is going to close, All right, this part you could put it activation. And then again, they're going to reset. Inactivation gate is going to open. So again, on the if the voltage changes, they can open up, and again, sodium can go in now. All right. So this is called absolute refractory period. Once they are inactivated, they're inactivated. It doesn't matter what type of voltage or how much voltage you use. They're not going to open up. Next up, we got the so again, in this picture is showing you where the absolute refractory period is going. This is when the sodium channels are start closing, the inactivation gates are closing, and basically no more action potential going in. But soon afterwards, all right, there is a relative refractory period. All right, so the relative refractory period, action potential might be positive. So at this point in time, sodium gates are closed and potassium gates are open and repolarization is occurring, but action potential could actually be positive because now is after the inactivation period. They haven't been fully reset, but if you apply enough voltage, that can get them open, and again, they could do that too. So action potential may be possible in this case, versus the absolute refractory period is not. Moving right along, interestingly enough, the diameter of the axon is important as well. In other words, the larger the diameter of the axon, the faster the impulses. Also, if they are heavily myelinated, then the impulses is going to be faster as well. So for that, we can go ahead and take a look at illustration to show this. If you have a nerve, for example, that it has a larger diameter of the axon. This is the axon, right? So we're going to pretend that you have such nerve, all right? And let's say they are myelinated. So these are the Schwann cells around it. Versus you have the axon the same length, but a smaller diameter right here. The diameter now is smaller, but the length is the same. So from here to here is the same, okay? And this one is myelinated too, all right? And now you have one that is not myelinated. Same length, same diameter as, let's put the number 1, number 2, number 3. And now let's go ahead and take a look at number 4. Has a smaller diameter, but no myelinated sheet, same length. And let's put number 5. Number 5, same diameter as number 4, but they're actually 2 neurons. If you measure these 2 neuron length together, is going to be equivalent to the same length as the top ones. And interestingly enough, if we go ahead and attach electrode to all of them and zap all of these at the same rate with positive 15, which is the minimum voltage required to get the channel, sodium channel open on the axon hillock, they're all going to fire action potential. But if you measure and figure out which one they're going to release the neurotransmitter first, it'd be the fastest one. This one is going to be the fastest, number 1, because the diameter is larger. Now, we don't want to just like memorize it. We want to know why a larger diameter could cause, to go faster. Action potential can go faster. Again, because they're a larger diameter, okay, they would have more surface area. And as a result of that, more of these sodium voltage gated channel. So in other words, more capability of sodium to go in to reach the threshold. In this case would be the negative 55, all right? So there are more channels are opening, is faster the conduction and so on. So basically, this one goes first. It will win, take the first stop, first prize. The second one, this is a smaller diameter, but still they have the myelinated sheets. So again, the propagation of these is referred to the action potential. So they go on, I'm sorry, saltatory conduction. So again, saltatory conduction happens in here. It insulates, it's create faster speed. So this one will get the second place, all right? Because it's identical to the top, except the diameter is small. Now this one is the diameter, number 3 is the same as number 2, but it doesn't have the myelinated. So there's no saltatory conduction. So action potential normally goes in, but you have to open up and close all those channels along the way rather than jumping. So therefore it's a little bit delay. So that one is gonna get 3rd place. The 4th place, they have less surface areas and so on, less channel on the surface area. So again, they're going to get 4th place. And the 5th place is the last one, number 5. They're going to release the neurotransmitter the latest. Why? Because there is a gap in here. It takes time for neurotransmitter to communicate with the receptor, then open up the channel, then ions go back and forth. So there is a little bit of delay. So this one we get the last spot. The last thing I would like to cover before I finish up this particular lecture is, again, this illustration of the saltatory conduction that we discussed. But there is an autoimmune disease known as multiple sclerosis or MS. This individual, they have an autoimmune disease. We don't know how and why. There are a lot of theories, which I'd rather not get into because that's going to make the discussion even lengthier. But our immune system cannot recognize our cells or the proteins that are on top of the surface and so on. So as a result of that, our immune system is attacking our cells, all right? In this case, the myelinated sheets are being attacked, all right, by our own immune system. So individual will have a lot of neurological problem because they're damaging the myelinated sheets. They are making them non-functional or sclerosis. And as a result of that, this individual is going to have serious neurological problem. There's no cure or anything for it. I mean, there are all kinds of immune suppressant drugs to slow down the process, but immune system continuously attacking our own cells. And that's called autoimmune disease. And multiple sclerosis is basically involved with the myelinated sheets that we discussed. You could see how, in this case, how fast is the tongue of chameleon, right? Or frog, for example. This is a slow motion, so you'll be able to see it. But this gives you an idea how fast the tongue is moving. The reason for it is the nerve that controlling this muscle, which is the tongue, right? It has a lot of those myelinated sheets that we discuss, all right? So the conduction of the signal in this case is super, super fast, all right? And again, is a communication between the nerve and the muscle. But, you know, the nerve, they need to get to where they want it to go. Action potential needs to fire and so on. So that's why. Anyways, I hope you guys enjoyed the lecture today. It literally took me about 3 hours with editing and putting the movies and so on. But the lecture, I think total came out to be a little bit shorter than that. But anyways, I hope you enjoy it. You could give me feedback a little bit later on, see how it worked out for you. To just mention and I will try to put it on an announcement as well. I found out one of the, it's not too bad. It's for the lab assignment, and that would be the action potential. It's already on the canvas. So after reviewing and mastering your lecture and reading the material, it's a good idea to do that, but it is set up to do it on Monday. But if you want to get a head start, that's fine with me. It's pretty intuitive. If you get a technical problem, you have to call the Labster technical support. I'm not going to be able to go over and troubleshoot, going over the troubleshooting with you, but it's pretty interactive. There's just one part that talk about the nearest equation, which we're not going to get into. So, but you could just manipulate the numbers and kind of like going through it. But the idea of movement of the ions and the process of action potential, I thought it would be pretty related to what we discussed, and it was a virtual lab. So again, keep in mind everything, it takes a lot of times. Be patient. I'll try to manage my time in a way to give you highest quality that I'm capable of doing. But keep in mind that you know, things we might have some hiccups along the way. There might be some problems along the way. You know, we just have to work together and hope that we can do this and we can learn and we can go on. Anyway, you guys enjoy the rest of the day, and you could look at it now as many times as possible. Talk to you guys soon.